He came here to listen to, first, Carol Cheah from the Ag Station, who is going to talk to us about biocontrols on Hemlock woolly adelgid and whatever other items inspire her. Carol?
Thank you. Thank you so much, Sarah. Is there a chance I could get a glass of water?
Yeah.
Thank you. Hi, everyone. Thank you very much. Thank you, Sarah, so much for inviting me. This is my first time to meet you all. I'm an avid gardener, so I actually feel very much at home with you all. I was the first postdoctoral student at the station. I worked with the esteemed Dr. Mark McClure, who was kind of known as the father of Hemlock woolly adelgid. He did all the initial research. And then I stayed on, and I was able to secure federal funding, actually, for my position. I work for the state, so I work for all of you, but I'm actually funded by the federal government. So I want to make sure I acknowledge the federal government for funding me all these years. So here's my little bio clip. I actually did this for University of Connecticut. We actually had a nice little collaboration meeting. They decided that we were in such similar areas and we really needed to coordinate better, get to know each other, and see what ways we could collaborate. There's different scientists from the different stations. So the Ag Station, we work for you directly. We do research that is to the benefit of the citizens of Connecticut, and obviously many times it is actually far beyond the borders of Connecticut. For example, Hemlock woolly adelgid is a national problem. But our first onus is to the citizens of Connecticut. So I have been at the station, as I wrote up there, for 24 years. This is entering my 25th year, and I've been working on Hemlock woolly adelgid. I also have a keen interest on elongate hemlock scale, and I'll tell you why in a little bit. Because in nature, no pest works in isolation. And that's what I'm going to try to convey to you today. Before I launch into the biological control itself, the candidate that we have proposed to be of use to the environment, I want you to understand the context of it, the context of the hemlock trees, the context of the ecosystem it is found in, and also all the abiotic pressures that impact the tree. Obviously, as gardeners, you all know soil is very important, water is very important, nutrients. So you put that into the context. You just can't, expect a biocontrol to be dropped in there and function fantastically in this huge forest. So that's what I'm going to try to convey to you today. So just in case you're new to this, which I'm sure you're not, biological control has a very standard definition. The father of biocontrol is DeBark. And so I just put his definition up there just to bring it into context. So many times you all know about invasive insects, invasive weeds, invasive plants, invasive pathogens that threaten our landscape, whether in the garden or beyond in nature. And why are they threats to our nature? It's because they've come here inadvertently or accidentally or even sometimes intentionally been introduced into an environment in which they have no natural control. So that is what biological control is. Biological control is a long-time concept. It was practiced way back hundreds and hundreds of years in China, actually. And so what you're trying to do is you're trying to bring back a balance to the system. Instead of having an invasive agent go out of control because it has no natural controls, what we seek to do as biocontrol protagonists are to bring in an agent that has been carefully researched. So let me stress that more than anything else. These are not ad hoc. I'm going to get something, it feeds on it, I'm going to release it. No, there are many, many steps in the procedure there before you can release a biocontrol agent. So I know everybody's very concerned about non-target impacts, but that is very much something that we consider when we do biocontrol, at least nowadays. And classical biological control is bringing the native natural enemy into the country of introduction of the pest. And other techniques that are actually very, very important too, and I'm sure as gardeners you understand this, is to conserve whatever natural enemies we have in place. Now, any of these insects or plants that come into our environment, they'll have some of our natives, will also feed on them, will also impact them. And as gardeners, we can do a great deal. Everybody knows about the crisis facing the honeybees and the pollinators. And believe it or not, many of the nectar sources that you provide in your garden can actually be of huge benefit to the beneficial insects that feed on the pests. So I'm a big proponent of that, of creating an environment where the biological controls can also flourish in times when their food is scarce. So that's the conservation of natural enemies and augmenting them. So just to show you very briefly, so now one of the biggest costs of biocontrol initially is the foreign exploration, because obviously you have to mount a whole research team, you have to have cooperation with the native country where this pest has been deemed to come from, and then you've got to go and explore there. So that involves setting up cooperative collaborations with the scientists of those countries. So that's a very big step. It has to be taken at the federal level usually. And then you look for the natural enemies there, then you have to quarantine and screen them. And that's where the whole topic of non-target impacts is very important. So you not only have to look at that, we'll take Hemlock woolly adelgid for the example. So you get a little beetle, say the beetle eats the adelgid, you see it out there, so you collect it. Now you got to test it not only on Hemlock woolly adelgid, but you're going to test it on any other insect that it might come across if you're going to introduce it. So you can see that this kind of screening can take a lot of time. It has to be done in a quarantine facility to start with, so it's lab. And as many of you know, labs don't translate into field. So then there has to be the field screening, etc. So you can see that's a very involved and a long procedure before you can bring that candidate to the federal authorities and say, hey, I think this is a great insect. I think it might help reduce the pest in the environment. And so then we go to this place here, permit for finding, for release, and that has to be determined by the federal government. So it's an environmental assessment. It has to be looked at by peers in the view. It has to be reviewed, and before they allow you to do a field release, you have to get this what called a finding of no significant impact. And then you come to the part where, okay, now I've got my permission, now I'm going to release this beetle, but hey, guess what? I only got 100 of them from, say, Japan or something. Now how am I going to combat this huge problem in the landscape or in nature? I mean, we're in the forest. I mean, the Hemlock woolly adelgid is by no means restricted to garden trees. So now we have to, the thing I put up there for mass rearing in red is because that is a huge component of biological control, which not many people are very aware of. Even scientists like myself we get excited about something, oh, it's a new candidate, it looks great, then how are you going to rear it? We can't go over to Japan or China or India or Europe and collect thousands of insects, bring them in with all kinds of pathogens. You have to be able to rear it. So that's why I bring that up. That's a huge component of biocontrol. You got to be able to rear the insect, and then even at that stage, you're releasing it and doing your environmental assessments of its impact. Okay, so just a little background there to show you that it's a very, very complex situation. I also work on MileaMinute biological control with Donna Ellis, which many of you may know her. She's a senior extension educator, a very great friend and a colleague of mine. I've worked with her for 9 years, and we work on MileaMinute biocontrol, and we are funded by USDA. Now, with that system, we don't do much research. It was all done for us. All we were brought on to do was to get the weevils. They're little tiny weevils, and they feed on this MileaMinute vine. And our task is to bring it into the environment, find the MileaMinute out there, find patches where we can establish the weevil and have it, and then follow its progress. So it's very much an implementation program. Now, my program here with Hemlock woolly adelgid was very far from that. It was right at the very beginning. I was brought in to look at the biocontrol agent that was brought in from Japan, assess whether it was any good. And then, that's how we started. And we had a big team. Obviously, I was not, I was just a little cog in the wheel. So I'm going to quickly go into the importance of eastern hemlock. Do many of you know the eastern hemlock? Okay, so I'm not going to run too much through it. Obviously, from a commercial point of view, hemlock is not an important commercial species because of its lumber quality. Now it's a very important ecological species. It's actually called, even some people call it a keystone species. It's a very old-lived, long-lived tree. It's been around for many, many millions of years. So that's just to show you some of the environment in which you might find hemlocks in nature. Very often around riparian areas, along lining streams, rivers, and down in the south, it actually exists as far south as Alabama. It's pretty interesting, but obviously it's so hot down there now. Hemlock, eastern hemlock, or Canadian hemlock is the other name. It likes cool, moist soils. So obviously, the further south you go, the higher the elevation it has to be. It needs that cooler temperature. So down in the mid-Atlantic and down into the Carolinas and Georgia, etc., it's only found up in the mountains naturally. So it has a very restricted and threatened habitat. Up here, I mean, you go up to Maine, we see hemlocks all along the coast, intertwined with balsam fir and other native trees. Here in Connecticut, we used to have a lot of hemlock down by the shore, but a lot of it was killed off. We'll go into that in a little bit. And so now most of the hemlock in the state is in the northwest corner, the big patches of hemlock in the northeast corner, and along the Connecticut River Valley, which again were killed off. Oh, I just wanted to mention, because you're gardeners, that hemlock is actually very popular, was very popular. I don't know if it still is, you can tell me. There are over 240 cultivars that I found. In fact, the American Conifer Society, I think, lists over 300. I'm not sure how many there are. There are weeping ones, prostrate ones, bicolored ones. It's a very beautiful plant. So just wanted to make sure that that came in it. So we have 4 North American native hemlock species. So eastern hemlock is the widest in range all across the eastern seaboard here into the Midwest. And it also has a small cousin, the Carolina hemlock. Anybody know Carolina hemlock? Yeah, it's a very restricted, it's a beautiful tree, but it's a very restricted. I've seen it in its natural habitat. It is actually very threatened by Hemlock woolly Delgid because it's a small endemic range down in the North Carolina mountains, probably in Georgia too. And then the other 2 species are western hemlock and mountain hemlock. And there is a Delgid associated with those, but now through genetic testing, they believe that a Delgid that occurs on those western species is endemic to them, so doesn't hurt them, doesn't threaten them. And we don't know, as for now, whether our Delgid over here has not been found out there, so that's a good thing. So that's just the background. When I first started doing this research, it was so impressive to me to see a giant hemlock. And so when I did some of the research, it was truly amazing. It's the state tree of Pennsylvania. It's very highly valued. I heard the term, I don't know where the term came from, but I've heard it called the redwood of the East, because the trees can be very, very old. Old growth hemlock is very, very minimal in Connecticut, as you know. I know of 3 spots where you can see some really big old trees. And basically, they were not cut down for charcoal or for clearing for agriculture because they were in inaccessible areas. But we really don't have much old growth hemlock left anymore. So this record material that I've found in the literature is from somewhere in Pennsylvania. They won't tell where it is in Pennsylvania. And I know there's some giants down in North Carolina as well. So just to give you a perspective here, this is a huge old tree, many hundreds of years old, and they've withstood many, many pressures from other pests and weather and other issues. So the other important thing about hemlocks, why we care so much about them, is because there's so many animal and species that are closely associated or even obligated to the ecosystem that the hemlocks anchor. And as I mentioned before, all these cold stream temperatures, you can't get the kind of temperatures in these streams with other vegetation. They've done studies where they've monitored water temperatures in streams that run in deciduous forests, in mixed conifer, and pine versus hemlock. And hemlocks have the deepest shade because they provide this eternal shade and this deep, cool forest conditions. And there are certain animals that need that conditions. And there are also many birds, because I love birds, and I don't know them enough, but it was very interesting to see the number of bird species that are very highly dependent on hemlock. So of course, the wildlife biologists and other nature lovers were very concerned when we had Hemlock woolly adelgid come in the scene, because there are all these bird species that are very dependent on hemlocks. And so I wanted to give a shout out. I have a wonderful cooperator. He's a retired professor. His name is John Weeks Jr., and he is a bird expert. So he is very kindly volunteering and doing some bird surveys for me in some of these affected hemlock stands where I've done the beetle releases. So we're hoping to see that, something's happened. We're hoping for the good here. And then this is a very interesting salamander. I've never seen it. Now I know about it. I have to get a picture of it for myself. It's called the northern spring salamander. And the DEEP, our Connecticut Department of Environmental Protection, has designated this as a threatened species because it needs that very cold, clear, clean water that only can happen in hemlock areas. So I actually have a site, that is the picture of my site there, where this salamander has been found. So you can see it has very wide-ranging impacts. The loss of the hemlocks is not just a niche for the wildlife. It's also there are a number of brook trout need these cold temperatures, and if you're a fisherman, you know how beautiful these brookies are. So that's just the background of it. And when I first started, of course, there were lots of hemlocks, and there was a lot of concern. Hemlock woolly adelgid was killing trees everywhere. And since I've been here for so long, I've found out that many growers have stopped growing hemlocks. Is that something that you've seen? I don't know. Whether it's easy for you to get hold of a hemlock anymore. I think there are very few growers that are actually growing hemlocks. And because of the pests that are associated with them, it's not so easy to order hemlocks anymore. I think when I looked for hemlocks for a project, as a student of mine was doing a project at UConn, and he had to order his hemlocks, I think, eventually from gardener nurseries, I believe it was. But we looked into Maine, we looked into Pennsylvania. So many commercial growers barely grow these species anymore, these hemlocks. We used to have a hemlock nursery in the state, but that got shut down too due to funding. So you can see, eastern hemlocks. And I'm here to give the message that it's not, you shouldn't be worried about growing hemlocks in your gardens, because they provide such wonderful shade. They're such a great hedging conifer. You can clip them and it would just provide, that's a beautiful hedge down there in New Haven, down there. And you can control, in a small setting, you can actually control Hemlock woolly adelgid and elongate hemlock scale with horticultural oil sprays. I'll show you that in a minute. So once again, where did this insect come from? Well, when Mark McClaw started working on it, the Delgid was first brought into the New Haven office of the Ag Station in 1985, in the New Haven office. And so Mark, who had already been doing a lot of research on Japanese insects, on scales and other insects, took up the challenge and he started looking at it. So in those days, we didn't have the genetic tools we have now. But he had seen Hemlock woolly adelgid in Japan, so he put two and two together and said he thought that it came from Japan. Now, since then, obviously, there's been so much more exploration. Hemlock woolly adelgid actually occurs all through Japan, China, even in Korea. I've never seen it, but apparently it occurs there. And it's in the Himalayas, and it's in the Pacific Northwest. But through the careful genetic investigations of Nathan Havel, who's now with the Forest Service in Hamden, he did all the collecting and did all the genetic stuff that he does, and determined that the adelgid we have here on the eastern seaboard is from Japan, which was what Mark thought all along, but he didn't have the tools to verify it. So what he did was he compared all this hemlock material, genetic material from all around, Hemlock woolly adelgid material from all around the world, and he found out that we have actually the southern Japanese strain of Hemlock woolly adelgid. And it was probably maybe one introduction, maybe it was several introductions, but it came from the same area. It's from southern Japan in the Osaka region, which is amazing. So, anyway, long story short, that's where we are. It's from Japan. Just so you know, I'll give you a little refresher. Some of you might know the adelgid from your own knowledge, but I'll just give you a quick primer on its life cycle, because in order to do any kind of biocontrol, to do any kind of scientific investigation, you really have to know your insect and its host, etc. So one thing, hemlock woolly adelgids are all females. They don't need any males to reproduce. They reproduce parthenogenetically. So you can see instantly there is a huge problem. You don't need to find a mate to produce eggs. So every single adelgid you see out there in the landscape is a female capable of producing from anything to 250 eggs. So right now you see the problem we have here. You know, it doesn't need a male. There are 2 generations. So right now we are in the 2nd generation here. So we are used to seeing these woolly masses on the foliage getting bigger and bigger during the winter. That's the long generation we call it cisterns generation. Winter generation is fine. And that generation is amazing to me because it's one of the few insects that actually feeds and develops in the winter. I don't know of any other insect that really is active during the winter. Most other adelgids all go dormant. There's balsam woolly adelgid, there's pine bark adelgid, there's larch adelgid. Adelgids are all common to conifers, but none of them feed in the winter, and this one does, and I'll tell you why. That's a really important thing. Just remember that. So winter feeding phase, winter feeding generation, and then right now we're in the summer phase. So the summer phase is short. So the eggs that were developing on the winter feeding insects hatched, now they're feeding, and we're going to get another generation a smaller generation of eggs soon. But we've had such crazy weather, and as with all insects, development, as with plants, as you know very well, temperature drives everything. The rate of growth, the rate of reproduction, the rate of development, everything. So we have crazy weather. I can't tell, now they are going to be just hatching. Nope, we don't know. We don't want to know the cumulative effect of all the cool spring that we had. So right now, this is the phase right now where we're getting some of these nymphs. And these are going to develop into adults and eggs, hatch. And then this is a very interesting part. I want you to hold, so you know, winter feeding. This is a very interesting phase that I'll bring up again later. This is what we call the summer dormant stage. So hemlock woolly adelgid, having come from a very mild coastal temperate climate in southern Japan, actually doesn't like hot summers. It goes dormant. Many of the adelgids actually go dormant for a short while, some of them for longer than others. And adelgid actually goes dormant basically from July till almost October. It is in this very inconspicuous stage right here, little specks with no wool, and so very easily missed. You might not even know you have hemlock woolly adelgid because you'd need to see those little specks. And I could tell you right now, my eyes are no good anymore. It's very hard for me to see this in the field without a hand lens. So this is a very key stage right here. And then in the fall, when it gets cooler and the photo period gets a little shorter, those adelgids that have settled on the foliage start to break dormancy, and they'll start spinning a little wool. So if you're a gardener or you're walking around the woods, the time that you will start seeing wool developing will be anywhere from October, November, December. By December, they should be larger wool tufts and should be more visible. And they're actually most visible during the winter because obviously leaves are off the other deciduous trees. All you have to do is look up into a hemlock tree and you'll see the white wool lining the stems. So that is the life cycle. Here just to show you what they look like. So in the middle of all that wool, so each woolly mass is a single insect, a single female. And if you were to part the wool, you can see all these eggs inside. And each of those eggs hatches out and becomes a crawler. Now this is the only mobile stage of the insect that's damaging. Because it has a wing stage, but the wing stage goes to a dead end and does not fly to anything and do anything. So this is the only stage that is actually mobile and is probably the one that's being carried around by animals, by man, by accidental introduction. So that's your crawler. That's the one when it settles down, it starts feeding. inserts it has a long feeding mouthpart, which inserts, it's very, very long, many, many more times the length of its body. It inserts that feeding mouthpart into the stem, and it sucks out the xylem parenchyma cells. So that's what it does. It's not a sap sucker. I know many people say, oh, it's like an aphid, it sucks the sap, but it doesn't actually do that. It actually targets the parenchyma, which are storage cells that enables the plant to put out new growth. So then it settles in there, it feeds. See, this is the little woolly stuff, and then it keeps growing, that woolly stuff. And when they molt, they actually come out of that wool, but you very rarely see it. And then they start spinning more wool again. They have little wax glands on the body. So that's kind of what it looks like. As I mentioned before, dispersal. And here's an interesting caveat. We actually do think that Hemlock woolly adelgid was accidentally introduced into the U.S. by a gardener. Funny. We all love exotic. I am no exception. I love exotic plants from other countries, especially there was a big rage for all the Japanese and Chinese species, that could grow here. We have very similar climates. And we believe that it was a private arboretum in Richmond, Virginia, that probably brought some Japanese hemlocks. And at the time, who's to know? It probably was in that dormant stage, tiny, didn't even look like it had an insect. And they believe now that they can pinpoint it to around 1951 or so. And it probably lay in that environment for a long time. It was an arboretum. It wasn't in the forest. It wasn't in the highlands or anything like that. It was fairly isolated. Richmond is in the lowland area. It's not like it's in a hemlock belt or anything. So it probably stayed there for a long, long time. So Hemlock woolly adelgid, as I told you, 1985 appeared in Connecticut. We've had it over 33 years. It started being a problem in probably the 70s, 80s in the mid-Atlantic states, but nobody took much notice. But this is an insect, nothing's going to happen. All of a sudden, it blew up and became a huge problem. So even when Mark McClure started working on it in 85, many people were like, it doesn't kill trees, it's nothing, it's just an insignificant pest. Boy, were they wrong. Boy, were they wrong. So as I mentioned before, it feeds on the parenchyma cells. What happens is then the tree is so susceptible to, and you can imagine this insect, because it can reproduce without males, it just covers the whole limb. It actually prevents the tree from putting out new growth. That's the primary thing you'll see. So needles when you get a badly infested tree, the first time it'll look a little sickly, a little chlorotic. But the major thing you'll see the next year is that branch that had all the adelgids on it will not be able to put out new growth. Now, there's no evidence that I have seen, even though it's been alluded to, that it has a toxic saliva. Another adelgid does that. It actually distorts the wood and the transpiration system in the tree. But to my knowledge, nobody's actually shown that there's a toxic saliva. I think it's just somehow the feeding prevents the tree from putting out new growth. It spreads over the tree, and because I believe hemlocks are so moisture sensitive, it causes this kind of physiological drought to the tree. The other thing I want you to take away also today is that hemlocks are also attacked by many other insects. And some of the most important insect is another invasive. I don't know if many of you know the elongate hemlock scale. It's actually not confined to hemlocks. It's called the elongate hemlock scale. It's again from Asia. But it actually goes on a bunch of different conifers. It goes on a lot of fir species are very susceptible to it too. Spruce gets it too. And currently, I think that is the greatest threat. But I'll go into that in a little bit. So just to know, we got two exotic pests on our hemlocks, and then we have the native pests. So what happened to our hemlocks before they even had these, in the last century or so, before they had Hemlock woolly adelgid, elongate hemlock scale, hemlocks had to fend off native attacks. So the native attackers were hemlock looper, which is a little caterpillar, and it just comes periodically, builds up, and defoliates trees, and then it kind of crashes. So it was not a consistent threat. Now we do have the hemlock borer, and that's a very interesting insect. That's what they usually call a secondary pest. If you all know about the emerald ash borer, well, the hemlock borer is a native related borer. And what happens is that this insect is specialized to take advantage of stressed trees. So what it does is it finds a stressed hemlock, invades it, lays its eggs in there, and then the larvae riddle their way all the way through the transpiration system under the bark there in the cambium and kills the tree. So, and I'll talk a little bit about that. So just bear that in mind. We had pests before, but obviously we've got now these new pests that are much more rapidly stressing and killing the trees. So I call it double trouble because many of our trees in the forest, and I'm just talking about the forest trees because that's where I work primarily, have both elongate hemlock scale and hemlock woolly adelgid for reasons that I'll bring to your notice. We now have very little adelgid in the landscape. I'll explain why. But we have a lot of scale, and it's advancing over much of our Connecticut forests. So what happens when you have the trees die? Obviously, it's very unsightly. It's terrible. You know, there's a danger. Trails are closed. There's logging. The MDC, the water authorities, have had to take down acres and acres. I was going to say hundreds of acres. It probably is hundreds of acres of hemlock around their reservoir systems because the trees were deteriorating so fast. And the other thing is that many of these forests, because hemlock wood deteriorates so rapidly once it is being killed by the insects, that if they want to salvage any money at all from the timber, they have to cut it while it's relatively healthy before it succumbs. And many of these natural resource departments are funded through the sale of their trees. It's a dichotomy, and it's a real pressure because they have to log and salvage trees in order to support their programs. It's not the best way. But that's what we have. And then you get, as I mentioned before, loss of habitat to the animal life that's dependent on hemlocks. And you also get nutrient losses, so erosion. Get a lot of erosion happening. And the loss of the hemlock canopy will bring in invasive plants. That's actually what we see. A lot of colonizing trees that come in when the hemlocks die, things like black birch, etc. But black birch is a native. It's not long-lived. It won't stay there very long. And then you'll see a lot of invasive insects move into those sunny patches where the trees have died. So you can see it is there's a long thing. There's a chain reaction, should we say, that happens. So here we go. Here's Richmond, Virginia, right down here where we think it was first started. Now I have a colleague of mine who's retired, and he's actively investigating the fact that he just doesn't think it's just one introduction. He thinks of the Gilded Age of gardening. He believes that because the popularity of Japanese gardens in those days, there had to be other estates that may have brought in hemlocks, Japanese hemlocks to the landscape because it was such a popular thing. So that'll be interesting. If any of you have ideas about, gilded estates which still have some hemlock component, it would be interesting to investigate that. So there we have 1997. This is shortly 10 years after we discovered it here in Connecticut. It was already widely spread. This is the very last map that I could get a hold of. And for some reason, the USDA Forest Service used to have a hemlock woolly adelgid site where you could go and download all the symposium material, that be postings from different states where hemlock woolly adelgid was found, and it's vanished. As of February this year, it's gone, and I don't know why. So that's the last map I have, which shows that it's about half the range. It's about half the range of hemlock, eastern hemlocks. It's all the way up into Maine, mid-coast Maine. It's all the way south, North Carolina, Georgia. It's in Kentucky. It's in Ohio. It's in Michigan. The latest discoveries, they thought, it was just, it was just somebody brought a tree in. No, it's established now in Ontario. It's established in Ontario. And the last year, this was very frightening. It was found in Nova Scotia. Now, initially, they thought, oh, it's just one of the, a bird brought it across, blah, blah, blah. Well, it was about 6 months ago, I was put in touch with somebody from Natural Resources Canada, and they did a flyover, and they found that 4 counties, or 4 provinces, I can't remember what the real terminology is, on the southern tip of Nova Scotia had hundreds of dying hemlocks. And so they're scrambling to see what they can do about it. I've offered them help. Obviously, we've dealt with 33 years here. Anything that they can learn from us, they're very welcome to. So that's the latest. Nova Scotia, way up there. So that's where we are. So as I mentioned before, 1985, it was found within 5 years or so. Look at that, by 1997, almost all the state, but by 1985, so I just colored it in to show you how it really went up went along the coast, went up the Connecticut River Valley, and the last places to get Hemlock woolly adelgid were the northeast and the northwest corners, where we have the most hemlock, of course. So I started looking into the role of temperatures and trying to understand why did Hemlock woolly adelgid expand so rapidly, We only came in with biocontrols about 10 years later, but by that time it was completely over all the state, most of the state. So I did a lot of research on that and took data, etc. And what this just shows you here is, I believe that it was our warmer climates, our warmer winters. Remember I told you it was a winter feeding insect? So if we didn't have periodic colds that might have killed off this insect, remember it came from southern Japan, it's not very cold tolerant. It was probably the expansion. This is the number of towns. There's a gap here because we had no records. So here's nothing reported, 1985. Then suddenly, boom, it rises up to like 100% in less than 10 years. It's kind of crazy. And the only thing I could think of is that it must be that the winters weren't cold enough, or they were just sporadic enough that it wasn't impeding the spread of the insect. So I want to tell you a little bit, I'm digressing, I know it's a long time to get to the woolly adelgid, but this is very important. My study now, I've done 15 years of data collection, and what I found is that we can explain why there are fluctuating levels of Hemlock woolly adelgid in the environment. It's because different levels of kill occur in the adelgid, and depending on where the adelgid has settled. So in the northwest corner, basically in the northwest corner, they're more cold tolerant, they've had more exposure to winter. It needs to be a much colder temperature to kill them. Along the coast, it's mild. We all know how mild it is along the coast. It's a whole different environment from northern Connecticut, and therefore a lesser cold snap will kill them too. But if we don't get that, we get this all over the place. So I just did north and south to just show you here. And these are levels of mortality, and I count thousands of adelgids. I've probably counted close to 250,000 adelgids since I began this study. And initially I had help, but in the last 10, 12 years, I've done them all myself. But it's yielded very good data. So you can see that we have had 4 episodes of very high mortality. See, that's 80% there. So this is 80 to 90%. So if you're seeing very little adelgid out there, I'll tell you it's because we've had dramatic winters. So in order to do this, just to tell you, so I do randomly go to the forest. I have selected sites I revisit every time, but obviously with the level of adelgid fluctuating all over the place, you can't always go back to the same site and collect, be certain of collecting enough adelgids to do the assessment. So basically, I'll go out there. I randomly find 10 trees, take an infested branch, bring them back into the lab, and I'll look at them under a microscope. I will pick at every single adelgid. And when they're dead, they are shriveled like this. If they're alive, they'll look plump and juicy, but you have to do to each one. You have to pull the wool away, poke them, etc. Oh, it's very tedious. And when you get to that point, you get a very simple proportion that have died. So I'll come up with an estimate for a site as a percentage of dead. And later on in the season, you will see this, you can see this yourself if you're familiar with adelgid. Later in the spring, say April, May, you'll see that, some of the adelgids never develop. They're all miserable, tiny things. And then you get one that's big, like that, and those are the survivors. So you can see how winter can actually remove a huge number of adelgids in the pressure, the winter one. But however, because there's lack of competition for the xylem parenchyma cells, the survivors can actually produce even bigger egg masses. So, you know, it's a little bit of a... But obviously, winter kill is good. Winter kill is very, very good. It's the best control out there. And so then I started to look at that. So we knew that winters occasionally happen. We had very cold winters in the 80s. I've been looking at all the weather data. But what was really important to me, and I'm actually really feel very privileged that it happened during my tenure here at the state, all these polar vortexes. I guess you've all heard about it. It's these abrupt plunging temperatures that came from the Arctic. And I have a whole other talk about that, how it's related to melting sea ice in the Arctic Circle, but we won't go into that. We'll just go into the effects of this polar vortex. Now, this polar vortex, because it's a winter-feeding insect, has a huge impact on Hemlock woolly adelgid. Many other insects are dormant. They've gone hiding. They're in hibernation. They're not going to be affected by these sudden cold snaps. But the Hemlock woolly adelgid is very affected by it. And the other way to understand this is to look at the climate divisions of Connecticut. I love my work because it really allows me to go beyond my insect. I love climate stuff, and so it's very interesting to me that the National Oceanic Atmospheric Organization designates climate divisions within the state. And you all know it, plant hardiness zones is another way of looking at it. But climate divisions is also with minimum winter temperatures, etc. So for me, it makes more sense to look at climate divisions because I'm interested in the minimum winter temperatures. And so, as I said before, Connecticut has these 3 divisions. Now, there used to be actually 5, but they decided it was too unwieldy. So they lumped everybody together in the central Connecticut into one division 2. And then coastal is 3. So when I started doing all my counts, etc., I was able to produce a mean mortality of the adelgid after each winter according to the division. And there were very clear patterns that emerged from that. See here, so in 2014, we had a very cold winter. It was in January. And here's what I got. So these are some of the results. These are the very minimum temperatures that it reached. In 15, we had a very, very long, cold, terribly cold winter. It was cold in January, it was cold in February, it was just prolonged cold. And this is the kind of mortality you saw. So after doing all that and doing some stats with the help of my more learned colleagues, I came up with some what I call predictors. There seem to be certain things you could tell, depending on the kind of winter. The minimum daily winter temperature was the best predictor. So if it went down to your absolute coldest temperature, so it went down to minus 20, you would be sure to get 90%. If it went down to minus 8, depended where you were. If you're by the coast, you'd get very high mortality. If you were further inland, So basically that's what it means. Just ignore all the rest of it. And just so you know, Division 1, which is the northwest corner, required the lowest temperatures to produce 90%, and it only needed to go to minus 6 by the shore. So that helped explain people like, why am my stuff alive? And of course, as gardeners, you all know, it's all about the microclimate. So obviously, I believe that landscape trees near urban areas, near your house, is going to be a little bit warmer, and you might get more protection. 2016 was the most wonderful data I could get because I was positive. All my data was showing that it had to be that minimum temperature that best predicted. So in 2016, people was like, what are you talking about? What do you mean the biggest mortality? It was a very warm year. I said, yes, it was. But we had one cold snap, and I bet you all know when it was. Mid-February, right? Valentine's Day. At my house, my generator failed. Everything failed. It was minus 22, I think it was. I'm from New Hartford. It was horrendous. Everything failed. It was just for a few hours. I got the data. It was basically from 4 a.m. to like 8 a.m. That's all it took. And we had the highest mortality for the whole state, 97%. And I've worked on this for years, and I've never seen a mean, an average that high. And it was because of that one. So that told me the minimum temperature was the most significant feature. Obviously, if you have a milder winter, but you have lots of cold periods, it's going to cumulatively kill the adelgid. And then we had this year. Now, this year was interesting because we had those horrible wind chills. I'm actually seeing damage to trees, which I think was from winter desiccation and not from the pest. Because now we've had 4 winters. Delgids are very depressed in the state, and I think this was an environmental stress. So you can see this is the 4th polar vortex that outbreak. I call it an outbreak because it's actually the polar vortex is the Arctic air around on the Arctic Circle, and what happens is that when you have an unstable jet stream, that cold air just comes down irregularly, unpredictably, and reaches into our lower latitudes, and that's what a polar vortex outbreak is. So in 2018, I did it again. I thought I'd done with all this, but I said, I gotta go and see what happened. So you can see now that the mean mortality in the northwest corner was not that great. I mean, it was great, but it wasn't 90%. Along the coast, it was so. If you live along the coast and you got Hemlock woolly adelgid, it might be time to do some remedial stuff. But it was very high in the northeast corner, and the wind chills were bad. So that's just the back of it. So now I'm sorry, I've taken a long time, but we're here to the biocontrol, because I want you to understand that when you do the releases, this is the background of what you're releasing into. You've got winter, unpredictable winters coming in, killing off your source of food, and even if it doesn't kill the beetle itself, it may be killing off its food source. And because it's a very specialist predator, how's it going to survive if it doesn't have its food? So I just want you to put that in the background. So this little beetle is a lady beetle. So now imagine putting these beetles out in the forest and trying to find them. It's like, just like give up. I mean, really? I don't think so. Anyway, just so you see, little specks, they're 2 millimeters in size. This is the adult, and they fly. They're very good little flyers. I'm very impressed with them. They live a long time, live over a year. And their larvae are small and gray, so they're not even very visible. So anyway, the most important thing I want to tell you is this is the only biocontrol hemlock predator that's available to the public. You could buy this beetle if you wanted to. And let me tell you how many years of research. I started in '94. I was brought in as the first postdoc. There was a huge boom. There's a lot of people working on Hemlock woolly adelgid biocontrol. They're working on other species. Why is it 20 years later we only have one that's available to the public? And I'll tell you why. Because you can rear this one. You can't rear the other ones easily. And I know because I was brought into some of these other projects to see if we could find a way to rear them. They're really hard. So this one is available because you can rear it. You have to rear it on Hemlock woolly adelgid, but you can rear it. And my friend Jamie Bonowitz, and I'll show you a picture of her, she is the person in charge of the rear. She worked on the rearing. I often help her with any tips that we try to come up with. So it's Tree Savers. The company is Tree Savers. They're out of a town north of Scranton, Pennsylvania, and they sell the beetles. And they're very dedicated. They're not out for money. They are totally doing it because she believes in biocontrol. They don't make money at all. They barely live by. But they're doing a great service to biocontrol by making it accessible to all you guys. I'm just going to take a little path down history here, because before I get to the numbers, this is the ag station where I work in. This is the Valley Lab in Windsor. We are multidisciplinary, but that is our station. That is where we rear 90% of our beetles that we release. We released 178,000 now, but most of it, over 95%, we released between 1995 and 2001. So I just want to show you, it's a bunker. It wasn't even a proper lab. It was a soil testing, leaching, experimental thing. We got a donation from a lady whose son tragically died. He was an arborist, Kenneth J. White. But because of her money, they renovated, we call it the Kenneth White Insectary. And really that's responsible for the way that we were able to rear these beetles. We had no funding, we had very little funding. and we rear them. And these are my colleagues from the 90s. It gives me great pleasure to see them. They're all gone on to better things, but that's Mark McClure there, Mary Kopecki, and this Beth Beebe. We worked very closely together, rearing these beetles, putting them out there, doing a lot of counts, etc. And then we had other folks there. So yes, that was where it started. Then I had no funding. I had sometimes funding, so I got sporadic help along the way, but never to the extent that we had in the 90s. So that's where we were. And then in the 90s, we put out so many beetles. I mean, we would choose to put out 10,000 beetles per site. We knew the enormity of the problem. The whole forest was infested. Even 10,000 beetles is hardly anything. That really is nothing. But we tried. We tried our best to put large numbers, so between 5 and 10,000. And then Tree Savers came along. And I was fumbling. I didn't have funding. I could barely keep myself employed. And Jamie was doing this thing. She's been in the business for 10 years. If you worked for somebody else before she became the owner of the insectary, then she was bought up. And now she has a little bit more stability. But Jamie is fantastic. They have this three people, maybe another seasonal person. Look at their operation there. It's in Pennsylvania. I went down to visit them last June. And they rear the beetles on foliage. You have to have Hemlock woolly Delgid at this point. I was working on an artificial diet on it, but we ran out of funding for that one. So they were very generous, I have to tell you. Last year, I approached them. I said, do you have any spare beetles by any chance? Because I really don't have any, and I would like to do some experiments now that the winter's killed everything off. Can we establish some new releases? She gave me 2,000 beetles. They're about $2 a pop. Yeah, she said, come on down. So I drove down there. She gave them. She gave them to me, packed up in a cooler, brought them in on the straw, and we took them out. And I did some experimental releases. So I'll show you a picture in the next slide. Last year we released 2,000 beetles, and I tried to target areas that hadn't been released before. And I also wanted to test their overwintering ability. So here we are. So you've got the winter context, killed off a lot of adelgid, 90% of adelgid. Now we should really use this opportunity to control and not allow that remaining surviving adelgid to expand and cause havoc again. So that's the whole premise. So I didn't approach her this year. She's been very busy this year. So I had a few beetles. So this is my latest little release. I love these guys. They're from Edgerton Park Conservancy Group in New Haven. It's a New Haven public park. It's a beautiful park. And they regularly spray their trees with horticultural oil. So I went down and said, come and see, our trees don't look good. I said, all right, I'll come down and look. And I'm like, you really don't even need to spray. You're spraying twice a year. There's no adelgid there. It's been killed off by the winter. So I said, at the most, you only need to do one spray. There's no adelgid. And they actually control the scale that way, too. Then we noticed that a couple of branches had been missed, and there was adelgid on them. Quite a bit, just a few. There were only 2 trees, really, that had. So I thought, oh, what a great opportunity. Let's see if this really works. I've got a few spare beetles, so we put the beetles out there at the end of May, and what we're going to see is whether those beetles could control that small unsprayed patch and prevent it from spreading over the whole landscape again. So they're going to hold off spraying for a year or so, and we're going to see how the beetles survive. And because it's New Haven and it's coastal, I'm pretty confident that they will survive there. So it's a new little experiment. So they will feed on other adelgids, but you have to have other adelgids there. So probably the most common adelgid that would be out there would be the pine bark adelgid. But pine bark adelgid is sporadic. It's not everywhere, obviously. So essentially no. Very sadly, essentially no. In many cases, there is no other adelgid that could fill the niche there. So last year we did all these releases. I approached state foresters. They told me of spots, and I went and looked in the forest. We found some infested trees. We did them at the fish hatchery. We did them in Burlington, Voluntown, Burlington. We went up to Roxbury. I was in contact at the land trust there, and we found trees there that were looking so sorry. They were infested with scale too. So I did a little trial release there at the Roxbury Land Trust. I also worked with the American Legion Forest. I worked with a small town park in Monroe. That's Dave Solek, a good friend of mine. It's a beautiful little park for camping. It's right on the Housatonic River. And I wanted to target areas I hadn't reached before. So we did 7 new sites last year. And I deliberately chose some very cold spots because I wanted to see if they can survive. So Great Mountain Forest was my coldest spot, and Coldbrook. So we're going to see. It's just the first year after the release. This is where we've released the beetles. As you can see, we've tried to cover the whole state. The big red dots are where the major releases were from 1995 to 2007. And the little yellow ones, and they really are little because they only released 300 per site, are the newer ones. Why I think this beetle is very good is because it is very host specific, so it's a very safe candidate. And it also is very synchronized with the adelgid. And one would expect that because it's a natural enemy from the country of origin. There are as another candidate also that comes from that region, but very hard to rear. So for those reasons, I've concentrated on this one. And the other thing is that all the stages of the beetle, whether it's the larva or the adult, they will feed on any stage of the adelgid out there, even that dormant stage. And to me, that's very important. That means no matter what the temperature does, it doesn't need the eggs to lay its eggs. It'll do it better if it has the eggs, but it'll limp along and lay on other developing. And so it's a very great flexibility in its predation. It's also got a very good lifetime fecundity. I followed beetles from maturity to death, and found out they can lay anything from 250 to 500 eggs, which is very compatible with the adelgid reproduction. And it has a very long season of predation. It starts a little bit more like mid-spring. It's not as cold active as some of the other candidates, but it'll start in mid-spring and it'll go all the way up into October. So the warmer we get, hey, those guys will still be out there chomping, which is why I like them. And the nice thing is it doesn't go into a reproductive diapause. What do I mean by reproductive diapause? Many insects that's only one phase in which they'll lay eggs, and then that's it. It doesn't matter if you provide the food. I worked with them, I know. They will not produce eggs until the next year. I'm like, no, this is not good. Well, this species will continually reproduce, so it's very amenable for mass rearing. And we believe, as a team, we did so much field work when we had a big team of about 3 or 4 of us. We were out there all the time checking, did they survive the winter? If they did, could we find them again? And we were pretty convinced from our early work that it established in the Connecticut landscape. It's just that now it's been many years and the trees are very big and the foliage you can't reach. Whether they're still there is a question to be solved, and I'm hoping I can find a way to do that. But it survived heat waves, drought, winters, everything. So I think it's a very good candidate. Just to show you, this is just to show you how the life cycle of the adelgid, there it comes out. It feeds on all the stages. It has a second generation too. None of the other candidates have a second generation. So it's able to produce a second generation on that second generation of the adelgid. And then they go and hide in winter. We're not really sure where. And so now we've got this, so we got this whole system. Now it's this crazy weather we're getting, cool summer, cool, wet, hot, dry, and droughts. I mean, what are these beetles going to do? Well, from what I've been trying to do in a controlled field experiment, it does look like they can survive our very hot winters, which is amazing. The humidity is so high. I just had some beetles outside. We had that blip on Monday. 90-something degrees. I'm in Windsor. It was so hot. I thought, all right, they're going to die. They're in little plastic cups. I'm like, okay, this is a very bad situation. I opened them up yesterday. Oh, they're still alive. That's good. That's good. So they come from a very, hot, humid environment in Japan, southern Japan, and they seem to be able to take our crazy weather. So in 2016, which was the hottest year globally, it was actually our second hottest year. The hottest year was summer was 2012, actually. I followed them outside in these cages, and I would count everything they ate. Yeah, it was very tedious. But the most wonderful thing was they survived the whole period, And I'll show you a slide as to what I came up with an average number they ate. Now, the other pressure that we have in our environment is the droughts. We just came out of a very significant drought. I'm sure all of you remember this in the gardens. And hemlocks are particularly sensitive because they're shallow rooted and they don't like hot, dry temperatures. It caused, I'm not going to say it didn't, it really caused a lot of stress to my forest plots. These are natural forest trees that don't get any additional precipitation. And I saw trees very, very stressed. And then, as I remember, I told you about the hemlock borer. We saw hemlock borer increase in outbreak out there too. But the beetles were fantastic. They kept feeding all the way through. And here's what I came up with for each. So I would follow individual beetles and actually literally count before and after to know how many they ate. They ate over 1,200 of those little dormant stages through the hottest time of the year into October. So I believe that they would be very good. I mean, if you had them out there, they could do a number on an infested tree, I think. It's hard to know how many you need to produce out there, but I think they could do a good job in a garden situation. And as I said before, the warmer the falls are and the longer the falls are, the beetles just keep feeding because I've been following them. And they'll keep feeding into December if they have to. But of course, they're a bit vulnerable. If you have a sudden cold snap, it might kill them or it might not. We have to do more research on that. The big overriding question is, can they survive our New England winters? We have some anecdotal evidence for that. I did a lot of work with Charlene Donahue, who's just retired from the Maine Forest Service back in 01-03. And I did my study up in Norfolk at Great Mountain Forest, one of our coldest spots, and she did hers in interior Maine, Quebec, Maine. And we were looking at Sasage's gymnast for another biocontrol, but we came up with some survival data. So -7, look at that. We had the same temperature both years, and the beetles survived very well. Now, they were caged outside onto the trunk of a tree. So it was a very different environment. They had nowhere to go. They had to stay there. They survived. And then the best part is that Colleen Tierling, who's now the entomologist with the Maine Forest Service working with Hemlock Delgids, she has given me tremendous data because they, with a bigger team, have gone out into their Sasage, Skimnasugi release sites along coastal Maine, all the way up to, I've forgotten the name of it, mid-coast Maine. And they've recovered the beetles. And this is natural releases. And I looked at their temperature data, and they got a lot colder winters, even along the coast. And they've routinely recovered the beetles, which is very promising, I think. It's obviously not a controlled experiment, but sometimes the best data is the one that you get naturally. So I think they have potential. They're adapting, obviously, to our New England weather. Now the problem is we're getting all these other abiotic pressures, the drought and all that. And now we can't do anything about that, obviously. And actually drought, they've pinpointed in the pollen record that the contraction of the eastern hemlock thousands of years ago, was due to a very long extended drought. So let's hope for more rain. We like rain. Drought also exacerbates the elongate hemlock scale impact. And that's what I've been seeing. However, I'm going to go to a nice, bright, happy thing. Before I get there, though, sadly, elongate hemlock scale is not so much impacted by the winter. You're probably asking, okay, so winter kills some Delgids. What about scale? No. Because the scale is dormant. It's dormant during the winter. This is even more tedious than looking at a Delgid because you have to actually pick apart each armored scale, pull out the insect to see if it's dead. This doesn't really show you very well, but what I found was that even if the female is dead, the eggs were viable. Oh, not good. So elongate hemlock scale is a huge problem. The borer, that's how small the borer is there. It's a native, but it will cause this. So if you see this during your hikes, please let me know. I'm trying to map where the outbreaks are occurring. What happens is that it's under the bark like this, it makes tunnels, and then the woodpeckers go after it. So just like emerald ash borer. This is actually the product of the woodpeckers stripping off the bark to get to the larvae, by which time the tree is dead anyway. but it gives us a very good visual as to know where there's a hemlock borer. And if you have trees in your gardens or landscapes you tend, the best thing you can do to help a tree during a prolonged drought is to give it some water. Just water around the base of the tree. Dirty water, any water. Just keep it mulched and cool, because that singularly alone can help a tree in extended drought periods. So this was so wonderful last year. We got some rains. Yes, I got to wrap up. So you can see the hemlock refoliation. This is a very wonderful thing. Trees are still alive out there. And just to show you, the growing stock in the state has not decreased. It should have gone down. If Hemlock woolly Delgid was killing all the trees, the predictions were so dire. But actually the growing stock, this is totally independent. It's nothing to do with me. Forest Service does this inventory. Our forest stock is maintaining itself. And whether it's due to the biocontrol acting together with weather, perhaps that's the whole thing. Just quickly, I'll leave that up for you. That's what the sites look at today. 70% of my sites where we did all the releases still look pretty good. And just last but not least, I just want to thank everybody, so I'll just leave that up there. And I'm sorry to take so long. Yes, I will repeat. Yes, and I'm going to be staying here so you can see me later too. Yes.
If you release, say, Pacific Forest or Beetle, are most lady beetles traveling elsewhere, or do they just stay there?
We believe they travel. Yeah. I'm sorry. The question was, if I release the beetles in, say, Bigelow Hollow State Park, would they be traveling? And I would say yes. And not directly from my experience. We found them about half a mile to a mile away right after the year after we released. So they were spreading out, but it's actually incidental anecdotal information from down south. They're finding them where they never released them. So we know they fly everywhere. Thank you. Yes. Okay, the question is, if we spray with horticultural oil, when should we do that? Generally, you can spray it any time of the year, but obviously you don't want to burn the foliage, so you kind of want to maximize. I would think early spring is a good time to do it. And you have to watch if you've got birds nesting. That could be an issue too. What you're doing in the oil is you're going to smother the insects, and it needs to be very thorough. So most people spray April, May, something like that. I think what you need to do is look at the tree and see how your Delgid is rebounding in the fall. If you see a lot of Delgid, yeah, you may need to spray again. But if you don't see much at all, and if you have a really severe winter, I would say don't even bother. And what I'm trying to do is set up a website at the station that I will post maps and give you an idea whether there's been a significant winter kill, and then you'll be able to judge whether you need to do a spray or not. And at that point, I think just one spray would be sufficient. And then you could do the biocontrol thing, maybe.
Would the spray kill the lady beetles as well?
That's a very good question, and I know I need to look at that. My feeling is that it may not so severely impact the adults because they could probably fly away, but I don't know if it would clog up their spiracles, their breathing holes. The larvae, they would, because they're soft-bodied insects, and they will suffocate just like the beetles. So ideally, it'd be better not to spray areas. If you released, say, your beetles on a couple of trees, but you have a big area, you have a forest stand or something, you could spray the other trees and hope that the beetles that you release the trees on will establish on those trees. But yeah, I think it would. Yes?
How big a problem do you think spruce spider mites are?
Spruce spider mites, they will come back. So neonicotinoids like safari and definitely imidacloprid has been known to exacerbate spruce spider mites. Now, spruce spider mites, I see it sporadically out there. It's not a huge problem in the forest. So, but I believe that with use of neonicotinoids like imidacloprid, you do get a rebound in spider mites. Yeah.
Carol, thank you very much.
Thank you.
Lisa Tewksbury is a research associate at the University of Rhode Island in the Department of Plant Science and Entomology. And she's going to be talking to us today about biological controls of swallowworts, and she may mention a few other things that she's doing as well. Lisa, welcome.
Worries me. Good morning. So, biological control of invasive plants, and specifically I want to start with biological control of swallowworts. This is the facility I mentioned that was built in 1994. And the reason that was built is that at the time we were doing biological control of insects, using insects, and we needed to do the part of it that's called foreign exploration. The example that I give is we were working on Colorado potato beetle. And actually before I came, my supervisor, Dick Casagrande, went with a number of people to Mexico to look for native natural enemies of the Colorado potato beetle, and they brought them back to a facility in Delaware. And with them being in Rhode Island and the insects being in Delaware, it was very hard to manage, to learn how to rear, to really, do anything with them. And so that's why the facility was built, so that we could be enabled to do good biological control work right in our own facility. At the time, it was very unusual for it to be in a university. Mostly they're USDA facilities. So ours is a small facility, but it allows us to do what we call classical biological control. And many of you might be familiar with the idea of, maybe putting a few extra ladybugs in your garden, or greenhouse growers who augment, what's going on in their greenhouse with some natural enemies. But classical biological control is a little more of an area-wide project where you say, in this area we have an invasive weed that's been introduced, or an introduced insect, and we would like to go back to the area of natural origin, the native area of this pest, and find out what's helping control it there. What are the natural enemies that are keeping it under control there? And so these are the steps that you have to go through. You locate what's going on there. I have a little bit of information about purple loosestrife. That's one of the first ones that I worked on, and they came up with 175 different insects that fed on purple loosestrife in Europe, and they had to kind of winnow their way down. So determine the impact. One of the things that you need to know is, well, you don't want to do a lot of research, spend 10, 12 years working on something that doesn't have a very good impact on the target, on the weed that you want to control. And then the other part that I'm sure you're aware of is the host specificity. If we're bringing something in for purple loosestrife, we don't want it to feed on some other plant that's growing in our wetland area. We only want it to feed on purple loosestrife. So the host specificity work is very critical. And then after you've done some of this work in Europe, you can bring the insects that you've decided to focus on into the United States and work on it. So I always want to, when I do this talk, give a little bit of the reasons that people are concerned about biocontrol, or the things that they worry about. We really don't understand why people ever used vertebrates for biological control. It wasn't a very good idea. And really, you don't want to use anything that's a generalist, so or that eats a broader range of things. One of the examples is gypsy moth biocontrol. A fly was brought in to help control the gypsy moth, and it turns out that it does also feed on some of these beautiful large butterflies. And the thing is, at the time they made the decision to do that, they knew it would do that. It was considered okay. So the changing standards is now we want to protect other insects, and at the time it wasn't really something, insects weren't considered, part of biodiversity, or biodiversity wasn't discussed then the way it is now. And so there's a similar situation with an insect that fed on thistle, and at the time, again, they knew that this would feed on other thistles, but there wasn't any understanding that anyone would want to protect a single thistle. They're spiny, they're horrible plants, and so why do we want to protect native thistles? So things have very much changed today, And biological control of weed researchers specifically are very careful about doing all of the testing that's required. And as I go through what we've done with swallowwort, I can give you a quick timeline that our graduate student came on board to do this work in 2005. He first started the foreign exploration in 2006 and brought back insects in 2006. In 2012, gave a talk titled Ready for Release, and we actually made our first release in 2017. So there we're on the 12-year plan. So this is what we have in Charlestown, Rhode Island, and that picture I actually took quite a few years ago. The site that I go to for collecting my plants to do the rearing on, and now I'm doing some research there as well, has been mowed pretty frequently now, so it doesn't generally ever get that bad. At the time when we found this site, it hadn't been bought by the conservation committee in town, and now it's used for hiking and for horses, and so they really are doing a pretty good job, you know, under certain conditions, about the best they can do, I guess is what I should say. So again, I mentioned black swallowwort is Vincetoxicum nigrum, and I want to give you a little bit of information about the naming. You will find, and you probably have used yourself, the naming Cynantium louisii. We use Vincetoxicum nigrum because it's a European plant, and that's what the Europeans use. I don't really know all the differences, and I feel like botany, the taxonomy changes a lot, but just so you understand, I do know that that name is out there. It's native to Spain, France, and Italy, and our graduate student did get to go there. The other target is this pale swallowwort, and the main difference between them, I didn't point out, I don't know if you can see the purple flower there. Not sure why it was called black swallowwort, if it was referring to the flower, maybe just because it's a very dark, deep color. And then the pale swallowwort is really pink. There's also a white swallowwort, and I mention that because the literature says that white swallowwort was introduced as well. All three of these are European plants, and it was recorded as being in New York, but I think at a botanical garden. And since then, no one else has mentioned it, except the last time I gave this talk, there was someone in Connecticut who thought they had white swallowwort. So we're going to follow up on that and find out if that's the case. But if you do happen to see a plant that looks very much like black or pale swallowwort but has white flowers, I'd be very interested to hear about it. So you can see, you know, with the picture of the black swallowwort, it was growing up trees. Here it's a huge monoculture. This plant has been spreading quite a bit throughout North America. This is the distribution of both plants, very much in the east, but you can see in Canada it's out in the west as well, and actually California for nigrum. The biology, it's a perennial, and it's in the milkweed family, Apocynaceae. So you'll notice that the seed pods are very similar looking to milkweed pods, and they do spread by wind, just like the milkweeds do. They're either insect or self-pollinated, and a very good invasive weed. The seed spread is good, but the rhizomes, well, the root system is amazing. And the way we reproduce it in our lab is just dig it up, divide it, divide it, divide it. You know, you could take cuttings, but we don't. And then whenever I've seen it mowed, the seedlings just come back in full force. So you can control it using herbicides and mowing, and there are actually a couple of good pest alerts, fact sheets online. One specifically written by Fran Lawler in New York, where she gives a good regimen if you really need to try to control it. So some of the ecological effects of swallowworts, endangered plants is one that people are concerned about in New York and Vermont and some other areas. We don't have any specifically that I know about. Milkweed is actually one that people are a little concerned about, with the monarch butterfly using it for its larval development. If this plant, when you see it growing in the pastures, it grows in the same areas. So this plant, by outcompeting it, could endanger milkweed for one thing. And then it does, cause a monoculture. And it also contains alkaloids, which are toxic to livestock. So that can be an issue. Again, just a couple pictures. The seed pods can be, very, very aggressive. Many in an area. We have been doing annual field monitoring to get some idea of number of stems, per area, number of seeds per area. And so we're hoping that once we get biological control off the ground, we'll be able to use those numbers to see if we're having any positive impact. So along with the just strict competition of swallowworts over milkweed, there also is a bit of a negative effect on the monarch directly. Because it's a close relative, the butterflies will lay a portion of their eggs on swallowwort. And then we've seen in the lab that those eggs hatch and the larvae start to feed and then they die. So the monarch can't use it as a host, but the female butterfly thinks that it is a host, and so will lay the eggs. So that's another justification for doing some control for this plant. This is just to show it also grows around it, can knock it down. There is a program called Monarch Joint Venture, and a number of USDA agencies have sort of signed up to help do a number of different things that will help increase milkweed, encourage people to plant milkweed seeds, and then our program is one on the list of things to do. As we went through our permitting procedure, we actually had a long, those number of years, there was a 3-year gap where we were waiting for Fish and Wildlife. And I found in this that Fish and Wildlife was one of these agencies that's associated with this Monarch Joint Venture, but we weren't really able to have that help us any moving things faster. So if you have a really small infestation, you probably can hand pick it, but anything other than just a few plants, I wouldn't think it would be very successful. So again, low densities, you could probably just hand remove it. Cutting does cause really vigorous regrowth. And grazing, I haven't heard anybody have too much success with grazing, again, because the animals avoid eating it. They know it's toxic. There is an area where they were trying to use goats to help control it, and the goats just would eat around it. It has a really strong smell. When you're out in a field full of swallowwort, it's a very strong smell. So I imagine animals, use their sense of smell as well. Chemical herbicides, as I said, can be used. And so because most of these controls are not either very successful or very long-lasting, or many people don't want to use herbicides. So we thought that it would be a good candidate for biological control. And actually, somewhere in the early 2000s, I had written a short paper for a biological control class just outlining that this would be a good candidate for biological control. But we wrote a number of grant proposals and wasn't able to get it funded. But the graduate student who came along, his name is actually Aaron Weed, who, I know, the best one was when my boss introduced him as, and here we have coming up to talk to you about swallowworts, a weed. So I don't know if that had anything to do with his interest. But so he jumped right on board and said, this is one that we should do, and contacted everyone in Europe who works with this plant or in the area of weeds, and really got it rolling. And so when he completed it in 2010, we carried on finishing the host specificity testing and all of that. So now I'm going to go through when this work was done in Europe. These are 3 of the insects that seemed right away to be good candidates. And one of them, the beetle, was a root feeder. The larvae are root feeders. But it fed on milkweed species, and so we had to eliminate it right away. Now, there are some researchers in Europe still working on it, because the initial testing that we do is basically a feed on it or starve and die. It's called a no-choice test. And so in that situation, they did feed on the milkweeds. In a field situation, it may be a little bit different. But since we had candidates that were very specific, we just moved along. But it is possible that some of the others may come up again later. So the 2 moths that you see there, the Abrostula and the Hypena, are the 2 that were specific. And we actually completed all the host specificity work on them, and we were ready to submit both to petition for release, but we decided to do one at a time, thinking it would be quicker and easier. And after it took about 5 years to go through that process, we really wished we'd made a different decision, because maybe now we'd have both completed. The reason we went for Hypena was because I heard Carol mentioning difficulty in rearing. A Brostula only has one generation a year, is very difficult to rear. Doesn't mate as well in our cages. And, but the Hypena has multiple generations a year, is much easier to rear, and so has a possibility to be produced for people to make releases. So this insect was found in Ukraine in 2005 in wooded ravines, and it was on pale swallowwort. And so you may hear, we discuss a little bit the idea of how well these insects are going to do in open sun areas, because it was found in shady ravines. But you can see there, let me see if I can, yeah, that's a large caterpillar that will be pupating soon. There's a tinier one there. It does some, the feeding initially is like little window painting when it's an early instar. And then later, when it's large, like this one, it will take chunks out of the side of the leaf. You'll also see, it's a little hard to tell, but the yellowing. One of the things we've noticed is that when there's feeding on the leaves of swallowwort by this insect, the plant starts to yellow. We think that maybe the plant defenses are sort of kicking in, maybe it's early senescence. And so one of the things, this is the second year that we're having our students do a little bit of work on the chemistry of the plant and the alkaloids and how the insect handles it. It just might help us with the releases if we know what's going on. And the plant does seem to respond differently to shade and sun conditions. So it's not surprising that the insect might as well. So I need to change this slide. I keep forgetting Noctuity. It's actually in the family Erebidae, if any of you are taxonomists. But it still is a night flying moth, and is only found from Ukraine, Turkey, Iran, small area. And until Aaron did his research, no one knew what the insect fed on. Someone must have had a light trap where they caught the moth or something, but they didn't know the plant it fed on. So that was something he was able to, put into the literature. It takes about 5 to 6 weeks to complete the development of the insect from eggs to new adult. And like I said, multiple generations. When we keep the day length and the temperature up high in our lab, we get 5, 6 generations. It's almost non-ending. And then it overwinters in the leaf litter or the soil. It has 5 instars. So we ended up testing 82. It was at least near 82. I've seen 79. I think maybe some we did lesser reps or something. 48 species of the family that milkweed is in, Apocynaceae. And that was, very deliberate, because that was one that we were most concerned about. And plants that grow in association with swallowwort. The other thing we did is the number of insects that we were interested in testing in the future. All of them have, what do I want to say? Close relatives. So Hypena, there are other Hypena species, like the hops looper is in the genus Hypena, and so that feeds on hops and a number of other things. So what we wanted to do is add to our list of test plants things like hops, or that Chrysalina beetle, the one I mentioned as being a root feeder. It has close relatives as well, and some of them feed on the Urticaceae, like nettles, the nettles. So we tested a bunch of nettles, which were not easy plants to work with. So the summary is that Hypena opulenta does have a restricted host range to the genus Vincetoxicum. So it will feed on the pale, the black, the white swallowwort, and possibly other swallowworts. Those are there's only 2 that we know that are in North America, the pale and the black swallowwort. Multiple generations a year. The larvae do target the young foliage first, and they will develop fairly well, whether it's pale or black swallowwort. They do just as well. And it does, the females produce a large number of eggs, and so they can reproduce pretty quickly. I just have a little information up here about the process of getting this approved. I said it took quite a few years. The first step is this technical advisory group, which is made up of people from a number of different agencies in North America. So it includes people from Canada, people from Mexico, because if we bring something into the US, it obviously will affect Canada and Mexico as well. It also includes people from Native American tribes, from Fish and Wildlife, as I mentioned. So the technical advisory group are scientists who read all of our data and decide if they think we've done a good job in deciding this. Because after their first read, they actually thought that we should test a few more things. And so we went back and added to the data. Then the second time I went to the meeting, and really everyone at that group agreed that the research was complete, that this was a very specific insect, and so they passed along the recommendation for approving a permit for this organism to USDA, PPQ, and everyone else that would go through the final process. So many of the organizations that are represented on TAG are also in the next steps. So it goes back through those actual agencies, not the TAG representatives. And so that's what took about 3 or 4 years to go through. And just a little bit about the membership. So USDA, NRCS, Weed Science, Corps of Engineers, all of those have members on TAG. So the regulatory issues that are involved is the Plant Protection Act says that our biological control agent must not be a plant pest. So if it feeds on a plant that we consider a pest, then it's not a plant pest. There's also the National Environmental Protection Act, can't be of any risk to the environment, and the Endangered Species Act. And it turns out that is the purview of the U.S. Fish and Wildlife Service. So that is what they were most concerned about, was would our insect have any strong impact on any endangered species. There are 1,500 insects on their endangered species list. So, that research has not been done. We have not been able to work with 1,500 insects to see if this has any, but we can go through the literature and we can go through the various ways. One of the things that they thought was something like the monarch butterfly takes the alkaloids from milkweed and sequesters them. And so, you probably remember from school seeing all the birds that will try to eat a monarch and then, basically vomit or something because it, tastes so bad. And they're supposed to learn, oh, that brightly colored thing, tastes bad. Well, these are cryptic. I don't know if you could tell that, but they're yellow and green, and they actually change color through their different instars, which I think probably maybe fits the growth of the plant. The lighter coloring of new foliage versus darker coloring. Anyway, the spotting, I'm not sure, but I do know that people who've been out in the field with them, they have been released in Canada, say they're very hard to find. They also have a, I call it spastic response. If you touch them, they do a, and fall off the leaf. So again, those two things are anti-predator responses that are different from the monarch. So we do not believe that this insect sequesters those alkaloids, and that's why we're sort of continuing the research to look at the chemistry of the plant and how it impacts the insect. So at the time we were going through this, there were a number of other petitions. Some of these you'd be very interested in. I'm trying to think. Knotweeds. Japanese knotweed is probably a favorite of many of you. And that actually has been approved. So there will be a biocontrol agent coming along for that. Air potato is kind of interesting, but it's only in Florida. So what happened was all the weed biocontrol researchers who were sort of being held up by this process wrote a letter of concern. And I think what they were saying is we've already devoted 10 years of our careers to this. Are we done? Is this, should we just stop working on it? Or are you going to find a way to move forward? Because if the regulatory process is not ever going to give another permit, then this work should stop. And so what ended up happening was the Fish and Wildlife Service and the USDA Plant Protection and Quarantine worked together, and now they hire one person who's 75% with one and 25% with the other. And so they can take all these permits and work through them, with both agencies involved, looking at what they both need to be concerned about. So that seems to have been working very well, and that was the result of us getting our permit in August of 2017. And just to go back to when the tag approval happened was in September of 2013. And I don't know the exact date, but 2 days later, Canada got their permit for release. And I sent them insects, and they released them in Canada. So we do have a little bit of information about how that's been going. That's the, let's see, the picture here. So 2013, right, 2014, and our release 2017. And there is one of the first Canadian releases. And you can see a little bit the green, like the yellowing of the plant. It's not quite as green. And so they're seeing that in the field as well. They've made 2 major releases, or 2 different areas. And in one of them, they keep seeing, the insect is established and it keeps coming back, and they're seeing some feeding damage. The other one doesn't seem to have been successful. I'm hoping to go up there in a couple of weeks and possibly collect some additional insects from the field that have been there for 3 or 4 years to kind of add to my colony. But we're going to be coordinating our releases and our monitoring methods to be able to follow this insect. Our first release site was out on Nishan Island, which is Elizabeth Island off Massachusetts. And there's a Nishan Trust that is very interested in doing biological control for this. They're interested in helping to manage as best they can all of the various invasives that they have on their island. They've used, goats was the one I mentioned. So this is the shady area there, and this is, the sunny area. And we're going to be releasing in both areas. There's the goats that they attempted. And I've seen goats used well for something like, knapweed for one. Maybe MileaMinute, I'm not sure. But not swallowwort. They really don't want to eat it. And so here's a cage, and we started out doing cage releases because of what happened in Canada. So I talked about how the caterpillars sort of wiggle away. Not only that, they really march away, or they can, to find better plants. We see them in our cages, when they're getting ready to pupate or whatever, they take off. The moth is a good flyer, and so when they did their releases and then moths emerged, they just couldn't find them anymore in that area. They might have flown miles away. And it's not that it still might not be successful biocontrol. I might release them here, and then you find them, 2 miles away, and they start up a population there. That's still going to be helpful. But for the person doing the research, you're not able to get your data. And so we set up these cages to be able to say, oh, they overwintered. And so we set up 4 cages on Nishan Island. They have both black and pale swallowwort. So in each of those areas, we picked a shady spot and a sunny spot because we wanted to see how well that did. And unfortunately, so far, we have not found anything emerging in any of those 4 cages. So we did that back in August and September. Now, similar insects are described as the adults flying in June. So we are getting near the end of June, but we're not quite at the end of June. So I'm maintaining a little bit of optimism. I will say that in Charlestown, we released, we only have black swallowwort there, so 2 cages, one in the sun, one in the shade. The shade had a moth emerge. And when it happened on June 1st, I was very thrilled, and I, in my mind saw a cage full of, 50 to 100 moths, and that has not materialized. So we are starting to, we actually set up a 3rd cage so that we could start fresh with a summer release. There could be something that we did wrong about our overwintering release. And so there's so many things involved. When we're getting ready to overwinter our insects, we put them in a colder chamber, and cool them down a bit and give them a shorter day length. And those are things that people research to really get right. So we might have had it not exactly right. So we're going to vary that a little bit. And we're planning on making additional releases. And this was inside the cage. We put them in there, they fed, they were happy. We will probably make a few straight field releases as well. But we want to be able to have a little bit of control over, keeping track of it, I guess. I'll do a little bit of Phragmites. You know something about that. Okay. I thought so, but I wasn't 100% sure. So one of the first things that happened when we started this project, we actually, right around the same time, we're doing lily leaf beetle biocontrol And then Sea Grant, Rhode Island Sea Grant, told us that we should do something about Phragmites biocontrol. And so we went to Europe to a place called CABI, where they have researchers who help do the foreign exploration part. If we're in the United States or some other country and our organism of interest, our target pest, is from Europe, they will do that foreign exploration. So we met with them both about our lily leaf beetle project and Phragmites. And we had done some research, knew that there were a lot of insects that feed on Phragmites in Europe, and in the US, all the information said nothing does. Nothing feeds on it. And once we started going out and looking ourselves, we found out that was not the case. We ended up finding about 25 insects, but in Europe, it's over 150. So this, though, is research by Kristen Saltonstall that she was doing right around the same time, early 2000s, and found out that some of the Phragmites that we have in North America is native to North America. Most of it that's invasive is native to Europe. And so that was able, that made us able to sort of target. We are targeting the European Phragmites that's invasive, and we don't want to target the native Phragmites. So more and more information has come out about the native, and people survey for it. In New York, side by side, there's exotic and native. The native tends to grow not quite as tall. It tends to have different color stems. It drops its leaf sheaths over the winter. You can find papers that go through. Here's a little bit of that here. And the native is not, fast growing, taking over. Now the problem is, I was at a meeting where I sat for the whole meeting listening to everyone talk about how horrible Phragmites was. And then Kristen got up and gave her talk. And right after that, it was like, we'll have to save our Phragmites. It's like, am I in the same meeting? I was just amazed at how quickly people could go from one to the other. They meant the native, not the European. But one of the curious things is a similar question came up when we did purple loosestrife biocontrol. I think Massachusetts was releasing an insect for purple loosestrife biocontrol. Rhode Island was, and Connecticut was saying no because we have an endangered plant, Lithromelatum, and we want to protect our Lithromelatum, and so we're not going to release this insect. Well, they ended up actually launching a major purple loosestrife biocontrol program when they realized that no one had seen Lithromelatum for many, many years. And so it's very possible that the purple loosestrife had outcompeted it. So that's one of the things that we're looking into and that people will kind of use as a decision-making tool. If we get to the point of getting a permit for an insect or it being decided if we should have a permit or not, one of it will come down to the native Phragmites and where it is and can we protect it. And so 26 herbivores, I said 25, only 4 of those are native insects. So the other 22 actually came here accidentally with the Phragmites. I think at ports of entry, when they do all the moving crates, if they pick it up from someplace in Europe with a big front-end loader or something that will pick up some of the frag as well and bring stems along. Many of these insects are inside the stem feeders, or the eggs will be on a stem, or on the underside of a leaf or something. And so easily these could be moved. So as we started doing our surveys, we found a number of insects that really didn't need a permit. They just came. So Arcanera geminipuncta is the first insect that we decided to work on. And it's a stem feeder. It lays its eggs on the underneath the leaf sheaths, and then the larva goes in, and actually I'll go to this next. It changes stems, so it could kill 3 or 4 stems. It goes through and it feeds to the growing point, and then it chews out, goes in, feeds again, chews out. Then when it's in its last stem and it's ready to pupate, it chews out a little window so that when it's a moth and it can't chew as well, it can push its way out. So it's kind of interesting. When they were doing all this work, they found out, some interesting insects live in Phragmites. There's one that almost makes like a little boat to kind of, move in the water from one stem to another. So we've been doing that testing, and we have found that Arcanera geminipuncta and Arcanera neurica, there's 2 species, are specific to the genus Phragmites. So that does include all of the native Phragmites. It will feed on the native Phragmites. We have uncovered a number of things that will make it so that we know that the insect doesn't prefer the native, but it is still, is still something that we'll call into question. Part of it is that the female will choose the European frag over the native. The next is that because the leaf sheaths drop, the eggs don't survive as well as they do on the European. So these are things that will, have to be worked out. We are writing the petition for release right now. So these are some of the differences in phenology. Earlier senescence, the leaf sheaths fall, and so we think that the eggs would be exposed to more extreme conditions. I was going to tell you a little bit about purple loosestrife, if you're interested. So how many of you might have been involved in purple loosestrife biocontrol? Oh, okay, good. I thought that might be the case. And so this is 3 to 6 feet tall. I swear that when I first went into purple loosestrife, it was 7, 8, 9, 10 feet tall. I mean, I'm 5 feet tall, so 6 feet tall wouldn't be that much over me, and I, could easily hide. 2.5 million seeds from one plant. I always thought that was just, ridiculous. Again, number of insects. Biocontrol researchers would love to find this sort of situation where you have an insect that feeds on seeds, an insect that feeds on roots, an insect that feeds on leaves. It sort of attacks all of the different components. What ended up working really well, and probably partly because it was easier to rear and easier to maintain, was this Galaricella. There's 2 species, Calmariensis and Pusilla, and they were often reared together. And so its immatures did most of the damage. And you can see on, this here, pretty good defoliation. So in Rhode Island, we had been asked to do this by somebody who was a horticulturalist at the zoo and said, I have a hard time getting native plants in our wetland walk because of this purple loosestrife. And I see online that there is a biocontrol program. And so this is where the plants were growing right out into the water. And that first picture is a July picture taken to show you the tall purple loosestrife filling the whole entire area. That was 1997, right around when we started releasing. This is a July picture in 1999, a couple years later, after we had 100% defoliation. And I took these pictures and sort of put them together to show that this is what it looked like. And then what was happening is the purple loosestrife didn't go away. One of the big questions is, well, what happens when it goes away? It didn't go away, but it kind of got smaller, and it started, it sent out branches. It wasn't, 8 feet tall. It was 3 feet tall and branching, and didn't produce as many seeds. And so you ended up seeing the native plants coming in here that the horticulturalist was hoping for, like arrow arum and marsh marigolds, and he had viburnums that he wanted, and anyway, a number of things were coming back. The interesting thing, though, is that this insect doesn't do as well in shady areas, and so basically we were very successful in getting the loosestrife out of the open water, but not as successful in keeping it, as managed in the shaded areas along the edges of the water. So so here's some of the plants: swamp rose mallow, slender blue flag iris. We actually released in other areas, but I've been following this for 20 years. And so when you talk about, the natural cycle of things, we really saw the cycle of this. So in a couple of years, the insect came in and had produced so many insects, so many adults flying around, that the zoo actually had to give refunds to a few people who didn't want to walk around a zoo with bugs flying around. It was only a couple of days. The thing I liked was that they said that it was related to fireflies. And as an entomologist, I was like, only because they're beetles, and like half of the insects are beetles, so it wasn't a very close relative. But it was just to tell people, I think, that it wouldn't bite or sting or anything like that. But after that, we saw everything good that you saw there. Somewhere around, so that was 1999 to about 2004 or 5, it was all looking wonderfully successful. Somewhere between 2005 and 2009, I thought, oh no, the loosestrife came back with a vengeance, and I didn't see very many beetles, and it really cycled back. Not the same as before. It never filled in the water area, but along the edges. And then again, somewhere around 2010, 11, 12, I went back, and it was completely defoliated. So I am actually thinking of taking some of my biocontrol students back this year to see where we're going. But throughout the state, we believe that the purple loosestrife is much more under control since we released the beetle. But it isn't in every single location. It works well in some and not in others. And I think I'm going to end with that. But I am happy to answer questions about these projects or biocontrol in general. The question is if I've had any experience with gout weed. No, I have not. Do you have a genus and species name on that? Agapodium? No. Egopodium? No. Sorry. Maybe not in Rhode Island yet. I don't know. Yes?
What promising work are you doing on that and stuff?
So it's a psyllid. I don't know if you're familiar with the type of insect. It's a sucking insect. And I'm not doing it myself as of this point. There is discussion, though, that this is Fritzi Grevstad. So if you look up, she's in Washington State. If you look that up, Grevstad, Fritzi, Grevstad. Though she was working also in conjunction with Roy Van Driesch in Massachusetts, and I'm trying to think who, I think someone in Canada. But they are either have their permit or are very close to it. And so there's some discussion in giving some insects to other labs to rear. And I'm hoping to be one of them. And in that case, I would, start letting people know about it. I don't really know too much more about it than that. Yeah, I have to say, I've mostly worked with either Lepidoptera species or Coleoptera, beetles, for biocontrol agents. I'm not very familiar with using a hemipteran like a psyllid, so it'll be interesting to see. But it is one that is certainly needs to be managed. No, yes?
Since the biological control of the Phragmites is not really established yet, what is the best scenario for
So the question is, since there's no biological control agent for Phragmites yet, what would you suggest as the best control? That's a hard question. Unfortunately, I don't do too much with control or herbicides. I do know that some people are doing things like painting stems, with herbicides, with the idea that it goes down into the root. That doesn't sound like, an easy task, especially if you have a huge area of Phragmites. But there are, even the University of Rhode Island has a class in invasive species management, or it's like a certification program. And they have various fact sheets, and I'm pretty sure they have one on Phragmites. So that might be helpful. But I'm sorry, it's very difficult. I know that. Yes?
When you do your research on candidate species, you look at the overall effect of the insect to the environment, say, are they being eaten by other things, and how does that affect toxicity or when you're not eating other pests like that?
Yes. And that is what the Fish and Wildlife would love for everyone to do, but they will not give all of the money necessary to do all of that. So I agree that would be great work to do, but doing the basic research, the funding isn't always there. So that's an interesting thing. I don't know how you get it so that the two things mesh, the research that we feel should be necessary before getting to that point and the actual funding, to do the research. We're doing the best we can. For example, that question about how an insect that we release impacts other insects that might eat it. That's why we're doing the chemistry work with our Hypena opulenta. And there are people doing. There's a Canadian researcher that's doing the same thing with one of her graduate students. So we believe that they, do not sequester, and so that it would not impact any insect that eats it. One of the things they were asking about bats, and I've never seen a bat eat an insect off a plant. They generally go for the flying insects. So that there's a number that we can eliminate for behavioral reasons, some for chemistry reasons, but It's a very good point, but as I said, we have 1,500 endangered species, so hopefully you could put them together into classes and groups to work that out. It is an area of research that I think is important to do, but I don't know that there's the support for that.
Have any of the biological controls that you developed been made available to the public for purchase? I get this question all the time.
Yeah, so the question is, are any of the biological control agents available to the public to purchase? She said she gets this question all the time. So yes, in many cases. So Galaricella for purple loosestrife, I think. There's various ways of being available. One is strictly.
I tell people to go for this purple loosestrife and see if they can find a larvae.
Yes. So Gail's comment is that if you are in an area where you know it has been released, but you don't see it in your area, you could go and collect, at a certain time and move it. For Galaricella, they were rearing it at the New Jersey Philippa Lampe lab, and they actually just stopped rearing that, and they're starting to rear Hypena opulenta. So that will be available. There are some commercial companies that will sell biocontrol agents, and it just takes time for them to, begin to develop it as something that people will purchase, I think, is part of it. I'm trying to think what would be other good examples. I know that I am working on releasing knapweed insects. I don't know if any of you have knapweed as an invasive in your area. They're spotted. We have black knapweed, and I'm getting it through the USDA. So in many cases, the USDA will provide it, especially if you had a group. Now, maybe an individual couldn't get it, but I get it because I'm a biocontrol researcher, but then I make it available to a group of landowners in Little Compton, Rhode Island. So those sorts of things could probably be done. So I would definitely, if you can't find it available commercially, I would talk to someone from either, who does biocontrol in Connecticut, like Donna Ellis, or somebody at the USDA, because sometimes they are available. Yes?
What about municipalities or states themselves with their properties where it's, these mass dragons are taking over? Are they willing to invest in it?
Yes. The question is, what about municipalities or state properties that are infested with these invasive weeds, for example? Are they willing to introduce biological control agents? And I would say yes, in many cases they are. I know that I was contacted by people from national parks who wanted to do purple loosestrife biological control. And I do find it sometimes easiest to work with either state or local properties. Like in Charlestown, it's the, it's a land trust or it's a town open space, basically. And so that is easier to get access, and it's good because it obviously that's property that's open for people's general recreation use, and so it's good to cut back, and then it brings it into the town and it will spread. So that's the thing about classical biological control, is even if you don't get a particular organism released into your property, the hope is that it reproduces on its own and it becomes a natural control, just as it would be in the native area of the pest. So Gail Reynolds and Donna Ellis have been working with us with lily leaf beetle biological control. And in New England, where we first released the insects that parasitize the lily leaf beetles, we would be working in individual little release sites. But now when people send us larvae from some other non-release site, we find that the parasitoid is there. So it has been spreading on its own, and that's the intent. Yep.
Have you heard any information about people having allergic reactions from handling swallowwort?
The question was, have you heard of people having allergic reactions from handling swallowwort? I have not. Have you?
Yes, I had some people come into an office and talk about getting skin rashes, and there was some banter on the internet about it. I don't know how reputable that was, but people were saying that a few people came and said they had skin reactions to it.
So I don't know if everyone heard that. He said there are people that have been complaining of skin reactions to swallowwort. I know that I've gotten hives and skin reactions when I'm out in a swallowwort field, but for me, I know I'm allergic to grasses, and there are tall seeding grasses, and so I put it down to that. But it's, it does have, like I said, alkaloids and other chemistry associated with it, so it's possible.
Thank you.
Yep. Anybody else? Well, thank you.